Here we go. All right, Jackie, go ahead and introduce our guest speaker.
Welcome, welcome. I want to first of all welcome all our students back to STEM Friday. Isn't this exciting? Our first STEM Friday for the fall. And we have an awesome guest with us today, Dr. Hawley. We want to welcome you. Dr. Hawley will be talking about nanoscience and nanoengineering. Dr. Hawley is a postdoctoral fellow in the Division of Neonatal Natology in the Department of Pediatrics at Johns Hopkins School of Medicine. Working in Lauren Jancy's lab, Haley studies non-invasive treatments for newborns with hydrocephalus, a fluid buildup in the brain. Motivated by improving children's health and outcomes, Dr. Hawley obtained a PhD from the University of Washington in chemical engineering, where she developed data science pipelines for analyzing shape changes in microglia, the brain's immune cells, as they respond to injury and disease. Due to her excellence in combining computational and wet lab research, MIT recognized her as a rising star in chemical engineering and the University of Michigan as a future leader in data science. Outside of research, Holly is passionate about teaching and mentorship. She's mentored over 14 students and developed a summer training program for students in Washington who want to learn about data science and research for neuroscience applications. We want to welcome you, Dr. Howley, and I will pass you the mic.
All right, so thank you so much. Hi, everybody. I am so excited to be here. I am okay if there are questions asked throughout. So whoever wants to handle that on the sensational side, I absolutely love questions. I will give a little bit of background to myself as well, since it might be a little bit confusing about how I worked in nanoscience and nanoengineering just from my introduction. I actually have some slides. I just got home from work and my cat normally gets a lot of attention when I just got home. So if she pops up, that's why. So my educational background is in chemical engineering, and yet most of what I do is studying health and cells. And so this is an image of a cell here, and this is a nucleus of a cell. And so I like to introduce myself through like this metaphor. And so if my career and how I approach research is a cell, then the nucleus of the cell, or really where all of that data comes from and all the information, is my training in chemical engineering. So chemical engineering is the discipline of engineering that is really focused on scaling up chemical processes. So a lot of students have to take chemistry in high school, and chemical engineers think a lot about how does something that a chemist creates on a small scale, such as one chemical, get scaled up to on millions and millions and millions of scales. You can think of it as something like the COVID vaccine or tests like that. How was it originally designed is one thing, but then how do you make millions of them and get them across the world? That's a chemical engineering problem. Um, so I got my bachelor's degree in 2018 from Arizona State University. So for those of you who are wondering, I graduated high school in 2014 and I got my PhD in 2023. So I'm almost a decade out of high school at this point before I got my doctorate. So at Arizona State University, I grew up in Arizona. I did a chemical engineering degree, and there I chose a really biochem focus. So which that means is that you can choose to focus on chemicals that are very synthetic. Like you can think of silicon is a chemical, all of those microchips that go into computers and that go onto medical equipment, or biochemical, which is everything that happens in your brain when you make decisions, in your muscles when you're moving, or even in like fermentation processes of food, or cooking is a chemical engineering problem. And there I also did an honors thesis. I'll talk a little bit about that and what that is. But then I moved to Seattle and I got my PhD in chemical engineering. And along the way, I also completed a certificate in data science. So I learned a lot of coding, and I apply a lot of coding into my work to understand what's happening in the real world. Some other things I have done, for those of you that have taken cell bio, this is an endoplasmic reticulum. My first time ever doing research was the summer after my freshman year the summer after my senior year of high school. And so I worked at a research institute and I studied how you can breed mice to express different genes. And that was my first time ever doing research. In undergrad, I did research for the first time in children's research. So I worked with Arizona State University Engineering and then the Phoenix Children's Hospital in order to come up with ways of detecting whether babies have diseases very, very, very early. Because the earlier you detect it, the sooner you can do procedures in order to help them. And that both got me very interested in neurodevelopment, which is why I'm working in the Department of Pediatrics now as an engineer. And it got me interested in engineering things on the nano scale. So engineering things that are very, very, very small that allow us to do cool things. And in my PhD, I also did some like cool things. I worked in silicon on actually making the wafers and chips that go into medical products. And I did a data science traineeship in national security, which was a completely different thing from what I did, but I got to learn lots of cool machine learning and all sorts of stuff like that. So that's some introduction about me. But I did my PhD in a lab that does what's called nanoformulation engineering. So I did my PhD in the Elizabeth Nance lab, or the disease-directed engineering lab at the University of Washington. And some of that work on nanoengineering is what I'm going to present on today. So when we study neurological disease, my applications as an engineer are the neurodevelopmental brain. So what that means is I'm very interested in the brain from birth to about like 3 to 7 years of age was what my PhD was focused on. Very early time in life, when all of your neurons are developing, you're making new connections, you're learning languages and how to move. And so there's many reasons to study neurological diseases. That's the time period I choose to study. And there are reasons like scientific discovery. What do you want to know? What can you learn mechanistically about how we learn or grow? Or we want to help people, like the lab that I came from is hoping to develop new drugs that can help treat diseases. And there's many different ways to study them. So this is an image here. This is a rainbow hippocampus. So this is a part of your brain, and all of these little dots are different cells, and they're very, very dense around these tracks, which make this like pretty butterfly type shape. But there are so many different ways that we can study diseases. We can study them in different models. So we can study diseases in cells that we grow on plates. We can study them in humans through clinical studies. They can be studied in mouse models, rat models, in so many different ways. And we can characterize those diseases in a ton of different ways as well. We can characterize the genetics of a patient to see if they have some mutation that causes a disease, like what I did in my undergrad. Or we can characterize a behavioral phenotype of a baby to see if maybe they're having developmental issues, such as, are they crying when they should be crying? Are they raising their head when they should be raising their head? Or we can go even deeper and we can take images of the brain. We can take images of cells. We can do blood analysis. Like there's so many different ways to get answers about biological issues. And there's so many different ways to image. Some of you all might be familiar with some things like an MRI, which are those huge machines that take these really cool images of the brain. or there's CAT scans, there's x-rays you have to get done at the dentist. And then on the preclinical side, where I've been, there's a ton of different types of microscopes. So what a lot of people are used to in science labs are these small light microscopes. But in my PhD, I worked with a microscope that was probably a couple $100,000 that uses very high intensity lasers to get very, very nice images of cells. And additionally, we have different targets for what we want to treat. So a therapeutic is some treatment that's supposed to make something better. And so you can think of this like when you use a Band-Aid that maybe has Neosporin, what you're targeting is a wound on the skin. But when we're treating neurodevelopmental diseases, we have to treat the brain. And there's different regions of the brain maybe we want to reach, or maybe it's a systemic problem. We have to think about questions such as, do we inject a drug? Do we inhale a drug? Do we have topical treatments? Does surgery need to be involved? Or if surgery does happen, is there a way we can apply drugs then? And then finally, this like nano side is nanotherapeutic formulations. How do we make them and how are they working? So something is considered a nanotherapeutic if it is on the nano scale. So we are all probably familiar with the centimeter. So a centimeter is 10 to the negative 2 of a meter, and a nano is 10 to the negative 9th. So it's 7 times smaller than that. And a nanotherapeutic formulation is anything that is on that scale of a size, very, very small, can't typically be seen with the naked eye, and needs to be imaged on a microscope. But complexity of biology doesn't surprise anyone. There's a ton of different researchers that study things. There are a lot of diseases that don't have cures. And it's actually a very general opening, I think, for a research talk, but I find it to be very interesting. I really like to combine a high-level approach to why I do what I do, which is to hopefully heal childhood brain diseases. And I'll get a little bit deeper into my specific topics. These are images of the brain's immune cells on the right here. They're pretty cool, I think. So in the NANS lab, I talked a little bit about it. We used these nanoparticle formulations. So they're these small circular particles that we made to probe and treat the diseased brain environments. What I mean by that is treat is probably what you think about a therapeutic. So you want to make someone healthy again, you want to treat whatever is making them sick. But we can also use these nanoparticles in order to learn more about the brain. So we can inject nanoparticles into brain slices, or we can study how they move throughout a brain to understand disease and spacing and things like that. For instance, this is that hippocampus we saw before, this really beautiful rainbow shape here. We zoomed in even further. This is work from my lab mates, and she was able in a publication to find where this specific drug with curcumin had been taken up into the brain. Here's a zoom out picture, and here's the images of some of the brain cells where there are these green blobs, and these blue blobs are nucleus of other cells around them, and we can study how those cells change and interact with different small drugs. So I developed a data management approach to nanoformulation. So what does this mean? So I am a data scientist, or a type of computational researcher on top of doing wet lab work. And so what data scientists do is that we use coding to explore more information about the real world. So it's a little bit different than a computer scientist whose job is often to code or to develop software or to improve software. Our job is to use code to understand science. So I did this thing, which is called developing a data management schema, where all of this pieces of information that can seem very, very spread apart, like whether something is tested in a human and how a behavior relates to genetics. And I used data science or coding and machine learning models and database management to create new roadmaps between them so that we can better understand how some behavior might be impacted by a therapeutic that we're developing in our lab. So in nanoengineering, basically, one of the easiest problems was that we had made these thousands of nanoformulations, so thousands of tiny therapeutics, but it was really, really difficult for us to understand how the way we made the therapeutic was impacting the biology of that we wanted to treat. Someone's drawing on the screen. Hi. So if you imagine these are little nanoparticles, something that we need to keep in mind are things like if they have a positive or a negative charge. So in physics, we have electrons and we have protons, and everything that exists has some type of charge or it's neutrally charged, and those charges balance. But if you are putting a drug into the human body, you need to be very aware of these positive and negative charges as they will interact with the body in different ways. And so there were current solutions to manage all these different formulations and how they interacted in the body, like reading what's in the literature, asking other lab mates. But I wanted a computational approach where if I had all of these different types of nanoformulations with all of these different types of charges, I could better predict which ones might be better outcomes. On a data science side, these things can be quite difficult as these projects span multiple universities, a ton of researchers, and all of the data doesn't always look the same. So on a computational side, the computational approach to research is to understand who has done all of that work, figure out where the data is, how that relates to reality, decide how we can standardize it, gather it up, and design some kind of database of nanoformulations. But I'm based in a chemical engineering department, and chemical engineers see things in process flow diagrams. So if you want to think about how I see my research, I see it as everyone that does research, like a scientist like me who's regularly in a lab coat, every person we collaborate, and then all of these nanoformulations that we engineer. This is a picture of a floor centrifuge. These are these super, super, super fast centrifuges that allow us to separate very tiny things. Ways of making nanoformulations, tons of really cool ways of characterizing what their charges are, what their pHs are, if they have any proteins attached to them, and a lot of different ways of measuring how these react inside of a brain. So with computational methods, I was able to create a roadmap. There's this really cool program that you can use through chart, and this converts it into a very code-based language. So I use a language that's called SQL to do this. And these are just more scientific IDs for everything I talked about. And I ran it on a cloud computing software. So basically, I took all of that information that went into making nanoformulations. So these are 4 methods of making nanoformulations. This one where we emulse, so we put a polymer inside of something else. We mix it up with a sonicator that runs at a certain energy level, but we can keep all of these variables in here and we can connect them via these things called keys to other pieces of information. The specifics don't really matter, but the biological applications, I've presented this before and that has always loaded before, so it's not loading on my screen, but the biological applications are what is really, really cool. So this is what like a day-to-day of nanoengineering looks like to me, which is I used a lot of code. So to give you some insight into what this code means, all of these are different scientific variables. So this is the nanoformulation that was created. These are informations about the researcher who created them. And these are informations about how big those particles are, how variable those particles are, I'll go into it, and what the positive or negative charge is. And then I was able to sort through these to find out cool scientific information. So this code just lets me select every variable, find all that information, and then define scientific constraints that matter. So this is why constraints like charge and size matter. So in your brain, if you want to deliver a drug, your entire brain is bounded by this thing called a blood-brain barrier. So all of these vessels run throughout your brain, and these blood vessels create a barrier, and they're supposed to keep bad things out. But occasionally we want to get drugs in, and that can be very, very hard. And then once we get a drug past the blood-brain barrier, we need to maybe enter these different areas, which is like extracellular space, the space between cells. Maybe that's where we want our drug to deliver. Or maybe we want to get into the neurons, the cells that fire electricity that let you move hands and your feet. Or maybe we want to get into the glia, which are all the other cells in your brain that do really cool things like create a conducive sheath that actually lets your neurons fire, or that respond to your immune system, or that really support how your neurons connect to one another when you learn a new language, or things like that. So I was able to query all these nanoformulations that are from my really cool lab mates, figure out how they were made and what's important. But there's other things that matter, such as nanoparticle size and zeta potential. So this is an image, it's a cartoon, but it's kind of what macrophages look like in your body. So macrophages are an immune cell. Whenever anything bad gets into your body, it's their job to eat it. So if nanoparticles are too large, they're bigger than 100 nanometers, a macrophage will eat it. If they're too negative, a macrophage will eat it before they ever reach your brain. So I use computational methods to narrow down the formulations we had created to ones that will not get eaten by a macrophage before the particle is able to make it through your bloodstream and into your brain. There's another one. There's another one. And then we can do things like check a PDI, or this is called a polydispersity index. So when you get a drug, oftentimes they come in a vial. What you want is not this. So this is a bunch of particles of different sizes, and this would be a very high polydispersity index because these ones are really, really small and these ones are really big. What you want is your drug to be very uniform in size, uniform in charge, so that what you're injecting is very similar and not so heterogeneous. So another thing I do that uses coding to impact real-world results is that we get rid of any nanoformulation that is too big. I don't know why that just swapped over. Anything that is just too dispersed is not uniform enough. And so this brought us down to a much smaller list that was interesting to study. So these are all different nano drugs that people have made in my lab and different features about them. And then when I actually picked some that were possibly interesting as a therapeutic effect, I was able to look up really cool things about them, such as this is a drug loaded with curcumin. A lot of people probably know curcumin as like a really healthy spice, but it can actually also decrease inflammation in the brain. And so some of my lab mates put curcumin into nanoparticles. They actually appeared, based on my query, to fit the constraints of what would be delivered into the brain and what would be beneficial. And then we can answer really cool questions like how many of the things that we made have the correct values to get into the brain. How can we use different materials? These are different chemicals that let us make drugs faster, better, cheaper, or let us make drugs that can actually fly from the US to different countries. That's important when you think of when COVID vaccines were trying to get to rural areas. A lot of times drugs need to be refrigerated. So how do you use computational methods to predict chemicals that'll keep things more stable to reach more people? But then I also wanted to expand this into how do the nanoformulations that appeared to look like they would work biologically actually work in biology? I showed this slide already. So this is an assay. So these are results and I'll explain them. So an LDH assay is a lactate dehydrogenase assay, or it detects how cells respond to some type of treatment. And these two plots are supposed to show you the same thing. So these are two nanoparticles that were applied in a biological system that looked the same based on standard ways of measuring them, but were made in different ways. And the scores don't matter so much, but you just know this is the cells lactate response after 1 day, 8 hours, 4 hours, and 2 hours. The plot should be identical because the nanoparticles looked identical, but we see that there's a very large difference in how the nanoparticles features, the size and polydispersity, impacted the biological system. And so I was able to use computation to show that we need new ways to characterize nanoparticle formulations to understand their impacts on cells in the brain. So that's where I kind of ended some of my work with the lab, but they're expanding the database further. We're trying new drugs. We're trying new capabilities. We've also been combining something that's called molecular dynamics, which is a computational method of anticipating physics of these nano-engineered relationships between proteins and drugs to predict better drugs that we might want to build or try. And then increasing the ability for other people to use this method to test their own system. And this is my PhD lab when I did this work, and everyone that's bolded submitted work to this group. So there's a little bit, I hope that was informative about how I use nanoengineering and computational methods to treat brain diseases. Any questions?
I'm going to open the chat.
I hope that, please ask if any part of it was unclear as well.
Yeah, I opened the chat, so anybody have a question that they'd rather put in the chat, they can do so right now.
And feel free to ask about any parts of it. I'm happy to talk about the neuroscience side, the cell side, the neurodevelopmental side, or even more about my career if anybody's interested there. I'm able to use code to help in real life treatment. So in my PhD, we were quite preclinical. So what that means is that we were very experimental. So we worked more on like cells than we worked on humans. Now that I'm in my postdoc, I'm actually a PhD that's working in a hospital. So most of the people I work with are doctors. And in this group, we actually get samples from humans. And so I am much more able to use my computational methods in a clinical environment. Those two areas have served different purposes. Like preclinical is a very great way to begin drug development, to explore some unique mechanisms. My lab was specifically developing these nanoformulations that 5-10 years down the road will hopefully be more useful to people. And now my current job at Johns Hopkins is much closer to the clinic, and our push is getting treatments as soon to patients as soon as possible. The second question, how do I use drugs to heal chemicals in red blood cells or replenish? So I'm not in red blood cell research, but I do work in immune cells. So I work in what's called microglia. So microglia are actually your brain's immune cells. And microglia and a lot of cells talk to each other through these things called cytokines, or they're just chemicals that the cells in your brain use to talk to each other. But oftentimes, when someone gets sick, their brain's immune cells send off a lot of these inflammatory signals. So you may have heard of inflammation. It's often like combined with like swelling or something like that. But what we can do is we can use drugs and therapeutics and other treatments, such as in babies, a really common treatment is cooling. So after certain diseases, they actually cool the baby's body down, which signals to these cells that they need to stop sending out these inflammatory signals because they can cause too much harm. A little bit of inflammation, though, is good. So it's a careful balance. I do not do red blood cell work, so I can't speak on replenishing them. And I talk more about my job. I'm going to get water. I will be right back.
So get your questions out. Start jotting down some mental questions for her.
Sorry, everyone. I talk a lot at work, and then I needed to take a break to drink. So my current job is I'm a postdoctoral researcher. So what that means is I was hired for this goal of supporting a grant that's in the Department of Pediatrics. So at Johns Hopkins, I'm in the Department of Pediatrics, and specifically within that, the Division of Neonatology. So neonatology is children younger than about 27 or 29 days old. So it's from birth to that old. So they're very, very young. And in that department, we have a few like clinical trials going on. We have a lot of collaborators, and it is my job to do a lot of methods of studying how already approved therapeutics work on treating the brain. So my day-to-day can look very, very different. Like today, I got in pretty early. I manage a team of researchers, so I checked in with them, and then I learned a new method to extract RNA. So that's some of the like DNA. It's a version of information storage in the body. So we can extract that out of blood, out of tissue. You can extract it out of different bodily fluids like saliva. And then we test it for certain proteins that are associated with disease or inflammation. Then in the afternoon, I trained a couple other researchers on how to do that, managed a couple projects. I have a lot of meetings with collaborators, stuff like that. In my PhD, my day-to-day job looked a lot like taking a lot of images of really cool cells, and I did a lot of coding to quantify how the shapes of those cells related to whether someone was getting better or they were getting worse. As a PhD and as a postdoctoral fellow, my days are very, very different depending on the experiments that my bosses are currently pushing or that I myself am leading under the guidance of my boss. Some days, some semesters I also teach, so sometimes teaching is fit into that equation. I hope that answers some of that. There's a bunch more questions. What made me want to start my career? Yeah, that's a great question. So in high school, I was interested in science. My father is an engineer, but he's an electrical engineer, so very different than mine. And so I went to Arizona State University because I grew up in Arizona, and that was the university that was nearby. And I knew I was interested in medicine. I knew I was interested in bio, so I originally chose biomedical engineering. But when I was in biomed, I you learn application first in biomedical engineering rather than science first, and I didn't really like that approach. It works for some people, it wasn't what interested me. So I switched to chemical engineering because I wanted to learn the science behind why chemical systems worked. So in chemical engineering, you learn everything from chemistry. I learned biochemistry, you learn physics, you take more math than like any other engineer. And you learn how to look at all of those chemicals in a larger unit. So you learn approaches called mass and material balances, where you can study how many chemicals there should be, how those have converted on a large scale to others, and how that's impacting a system. I thought in undergrad, I wanted to go to med school. So I was pre-med until my senior year. I even took the MCAT that I'm sure some of you have heard of. But I was really unhappy when I was studying for the MCAT and studying for medical school. And I was doing undergraduate research, and my PI said, there's a student chemical engineering conference. You should go, see if you enjoy it. And I went there, and there was a panel of PhDs talking about off-target effects of nano formulations, just like this work. And I just knew I wanted to do it. I knew I had to do it. I came back to Arizona. PhD applications were due in less than a month. And I needed 3 letters of rec, and it was a Friday. And I told my PI I was going to take a gap year. And he was like, well, just walk and see if you get your letters now. And somehow all three of my letters of rec were still there on a Friday afternoon with their doors open. And someone told me to apply to University of Washington, that it was a good school. I'd never been to Seattle, so I applied. And then I found out when I got the offer letter that PhDs are paid for, unlike med school. I was expecting to take out a ton of loans because that's what everyone tells you to do for med school. But my PhD had a salary of $37K and I didn't pay any tuition. And that's how I started my route to becoming a doctor. In my PhD, I picked my lab because my PI, Elizabeth Nance, really cared about making a difference in children's brain research. And so I went there because she was a great mentor. And I had really fantastic mentors that encouraged me to do coding, that encouraged me to do all of these things. And I learned I would really love to be a professor one day while I was in my PhD. And so that's why I'm now at Hopkins, learning the extra skills I need to lead a lab of my own treating brain disease, and aiming to apply to become a professor in a few years. So that's kind of how it worked out. And I love it. I genuinely love my job. Okay. Someone asked, so a brain will send a stimuli reaction to other places of the body to maintain homeostasis. So some of your brain is focused on homeostasis. So part of your brain's job is to make sure your body is at the right temperature, to make sure that things are doing what they're doing. Your brain, your central nervous system and your peripheral nervous system, so nerves outside of that main spinal cord brain axes, are constantly talking to each other. Your cells in your body are constantly talking to each other. They do want to maintain homeostasis. So there's certain things you need to live. There's certain salts you need. There's certain pH levels that you need. You need sleep. All of those things are important. So your body is sending signals to yourself, but it also sends stimuli to places to get you to move your hand and open it like this to tell you that a pan is hot. There's so many things that it can do. And if you get into the realm of mental health as well, your brain tells you things like you've pushed yourself too hard, you need to take a rest, you maybe should go talk to some other people, things like that. Your brain is really amazing. Yeah, it's really cool. It's a really cool part of your body to study. How common is my job? Yeah, so that's a really great question. So there's a lot of PhDs out there. I'm not sure how many that number actually is, but it is a common path to get a PhD and then to become a postdoctoral fellow. That's pretty common. In engineering, the most common thing is to get a PhD and then to go into industry and work in a research type job. So if I had gone into industry to work at a company such as Boeing or such as Moderna with only a bachelor's degree, I likely wouldn't have been on the research side. I likely would have been improving processes, figuring out when things get wrong, making equipment more efficient. But with a PhD, you're able to be much more on the project planning, project exploration side of things. That being said, postdoctoral fellows are pretty common, but for an engineer to do a postdoc in a clinical lab is less common. That is pretty uncommon. And it is a path that my PhD advisor did. And so that kind of paved the way for me to do it. And it's also a path that has been enabled because I have sought out a lot of opportunities with hospitals and with clinicians to learn more of the medicine that I need to engineer systems that are relevant. But you can absolutely get a postdoctoral position with a PhD. You have to have one because you have to have an MD or a PhD. You have to have gotten a doctorate to have a postdoctoral fellowship. But what's really, really cool about that is once you have a PhD, it becomes less about the specific field you're in and more about what you want to research. So I'd been studying the brain for 5 years, but I'd never worked with a neuroscientist. So now my boss is a neuroscientist. Her closest collaborator is a neurosurgeon. And the people that work down the hall for us are fantastic pediatricians and researchers. And it's just an incredibly cool environment to work in. Someone asked, you have to plant the drug in the right area of the person, otherwise they'll die. No, I don't think we ever want to be at the point where if we put a drug in the wrong spot, they won't live. But a lot of times when you're talking about treating a disease, people are sick. That sickness does not always necessarily mean death. So for the case of studying neurodevelopment, some babies are really, really sick. And sometimes for that, you would need to develop treatments that would save their lives. In a lot of cases, it's not so much that without treatment, people will die, but without treatment, people will have significant impacts on their lives. They'll have significant social impairment or significant motor impairment, significant inability to communicate the way that is seen as efficient and standard. And so a lot of times that's what we're trying to improve when we talk about neurodevelopmental outcomes, are longitudinal, so over your lifetime, quality of life adjustments. What encouraged me to start learning about chemical engineering? Did I want to do this as a kid? No, I had no idea what chemical engineering was as a kid. Even when I was in a chemical engineering program, which a college advisor had convinced me to be into, the first day of class, the professor asked what chemical engineers do, and I couldn't answer the question. Like, I was in an introduction to chemical engineering class, and I didn't know what chemies did. And I didn't really get what a chemie did probably until I was graduating with my chemical engineering degree. When I was a kid, I loved, and this is still true, animals. I loved animals. I loved learning about different types of animals. I loved like going to the vet and learning how they treated those types of things. And then as I went through high school, I became much more interested in human medicine. And that's how I ended up where I'm at. How do you make drugs go through the blood-brain barrier and into certain areas like glia or the nervous system? Yes, so there's a bunch of different approaches, and these are huge fields of research. So there are a bunch of different ways that you can give someone a drug. You can inject a drug into the bloodstream with a shot. And even then, there are a bunch of different ways you can inject a drug into a bloodstream, and that's a genuine consideration for drug delivery. You can orally consume a drug, such as like vitamins, Tylenol, ibuprofen. All of those are an orally delivered drug. So you take it and then it dissolves in your stomach and in your intestines, and then you get the drug. How do we get it to the blood-brain barrier? Oh, there's more methods. You can actually inhale a drug, and then it's a mucosal barrier instead of a blood barrier. So that's another interesting one that a lot of people are pursuing. And how do you get them into glia or the nervous system is that you control those scientific and chemical aspects of the drug. So it has to have these characteristics that allow it to pass into certain systems. So there's a ton of science about like what chemical characteristics makes a drug good for the blood. And then once it reaches the brain, so once it goes and circulates through your body, it has to have certain characteristics in order to get to the brain. Someone asked a question about testing on animals. There is a lot of research that does involve animal research, but there's also a ton of research that we've worked in that, and this isn't necessarily about this isn't about my lab. There are a lot of labs in the world that do research on different types of animals, but there is increasingly more labs in the world that do work on cells. So I talked about some work on cells in this lab. So you can grow in this presentation. So you can grow cell culture, or you can grow cells on plates. So these are cells that are extracted. They can be extracted from the bloodstream. They can be grown in what are called immortal cell lines. There's a lot of work on that. There's a lot of work that makes it into press on what are organoids. So that's growing these cells into 3D structure. And there are a lot of human studies as well that can be done on things like blood samples and types of stuff like that. So I do a lot of computational research. And so what I work on is a lot of images taken of just these different types of cellular systems, images taken of brains, such as MRIs or images of human brains, those types of things. Do I test on live humans? I do not have an MD, so I am not allowed to test on live humans. Because these questions are going in a couple ways. I do not test my drugs on anything. I am a computational researcher, and I do some wet lab in characterization. I test how the drugs are made themselves, and I predict how that might work in biological systems by studying how that works in isolated pieces of biology. So we understand that a brain has a certain salt concentration. So like my first year of my PhD, I would take those nanoformulations and I would put them into a solution that's very similar to your cerebral spinal fluid. So it's water with the correct balance of salts that match cerebral spinal fluid. And then I would change that salt concentration in the same way that it might change in disease. And I would see how these nanoparticles changed. So I do not test on live humans. Testing on live humans is called a clinical trial, and it is very, very regulated, and it has to be done by MDs. So I don't have any of the credentials to do those types of things. How long have I been doing this job for? I started my job at Hopkins a month ago. I was in my PhD for 5 years. What college did I go to? I went to Arizona State University. The slide has popped back up. How many people usually decide to do my job? I would say at this point in my career, I am one of the only people in the world that has a chemical engineering degree that is working in a neonatal lab. That's something that is really, really cool about getting a PhD, is that you can really pave your own path. After this, I hope to become a professor, and there aren't that many professors in the world, and lead my own lab. So not a ton, not a ton of people. Someone just said, cool, thanks. What made me choose this career, I talked a little bit about, but it's followed up by, I appreciate your work in the neonatal area. So I'll answer a little bit about that now. So one of my first experiences was working in Alzheimer's. So I worked in a retired community in Arizona, and in that community, there were a lot of brains donated to science, and a lot of the research there was Alzheimer's research. And Alzheimer's research is a really big research field, but it is a research field that has had a lot of money and a lot of time that has not had a major breakthrough in a while. They had some drugs recently that have gone through, and I'm not caught up on the field to make commentary on that. But at the point that I was at, which was like 2015, they hadn't had a major breakthrough in a while. And then I came to the realization that if I really wanted to understand what happened later in life, I would like to understand how the brain develops in the first place. And that's also supported by scientific findings that injury, disease, infection early in life is correlated with earlier and higher instances of neurodegeneration type things. But additionally, neurodevelopmental and neonatal research are underserved areas of research. And so what that means is that most people and most pharmaceutical companies target the adult brain or the aging brain. And there's many reasons for this, But one of the reasons is that babies' brains are not profitable. So the brain changes so quickly when you're young that you can't come up with a drug that's going to treat a 1-week-old the same way a 1-month-old or a 1-year-old are going to react, because there's so much growth that's happening in that time. And so I was very, very interested in a field that is scientifically years behind adult research and geriatric research. Additionally, most drugs for babies are just scaled down drugs for adults. So they'll just take the weight of the baby and the weight of the adult, and they'll divide the amount of drug that's given. But that's completely wrong, since babies and children have very different physiologies than adults, which is, I think, very fascinating. So that's why I continue in the neonatal area. And I genuinely believe if we can understand neurodevelopmental areas better, and we can treat more neurodevelopmental diseases, not just in the neonates, but all the way through young adulthood, that we will have significantly less adult and geriatric degeneration. Do I develop treatment when my patients are ill, or do I order it and prescribe it? I am a PhD. I am not an MD, so I do not have patients, and I am not legally allowed to treat patients. And I am currently a doctor working in a hospital at Hopkins. But if anyone asks me to help resuscitate anyone, I am legally obligated to say that I do not have that type of training and that I am a PhD. So I can't speak on that. Is the work environment fun, or is it serious? I think it is quite fun. It is very serious in that we don't goof around all day, and we have a goal, and that's to treat babies, and we have deliverables, and I need to get things done. But it is very fun in that the pediatric team is made up of very good people who genuinely want to make these lives better. And wet lab, experimental work, computational research on large collaborative teams necessitates a lot of people. And so I get to talk to a lot of really cool people, and I get to learn from a lot of really cool people, and they've become some of my best friends in the world. I recommend getting a career in chemical engineering. Do you recommend going for it? I absolutely recommend going for a career in chemical engineering. It is the highest median salary of all the bachelor's degrees. So if you stop at a bachelor's degree and you go into chemical engineering, you are guaranteed the highest salary without needing any more education. This is often not really known. It is also one of the hardest undergraduate degrees. So be prepared for that. It has the highest high school GPAs, SATs, and ACT scores going in, and the lowest GPAs coming out. It is very difficult. It will make you a better student. It will make you better professionally, and you will really, really know your science coming out of it, but it's hard. But that being said, that doesn't mean you shouldn't go for it, because I genuinely think you can do anything. My friends in chemical engineering work in makeup. I had a friend that studied how lipsticks smear to come up with better products. I study the brain. My friends study batteries. Some people study solar energy. There's professors studying how do we recycle and digest plastic in a way that's sustainable for the earth. Genuinely, chemies can do anything. Yes, the geriatric brain neurodegeneration is also very, very fascinating. And we just don't understand so many mechanisms of why it is. I'm out of questions, so I'll kind of end with this, but I find it so fascinating that our brains are so evolved that we can use our brains to study brains, and yet we don't know everything about the brain. So yeah, that's kind of my job and what I do. Thank you, everybody.
Wow, that was great. That was a lot of questions also. I'm proud of you guys. And really excellent questions from Suzanne Middle School students here. Anybody
Thank
else
you
have
so much.
any, we got time for one or two more. All right. All right, Dr. Hawley, we appreciate you for
Thank
coming
you
to
so
speak with.
much.
Hopefully you'll come on our rotation because now you're one of us. And it was an excellent presentation, and I love hearing about all this new science and all these new procedures coming out where we feel like the first ones in line to know everything about it. We thank you. It was excellent. You did an amazing job, and the kids love you. So
Thank you so much. Those were great questions, everybody. I would love to come back. Email me again next time you need someone.
yes, we will. We will have you on rotation. We usually catch up with you again in about 6 months. So stay tuned. And
Okay.
you guys, I took your name down for your